All right, so let's go back and let's go back to where we're at. So here's what I really think you should do sometime before Tuesday is to go back through the chapter and actually look at it and maybe read it and actually say, I'm going to look at that example, I'm going to look at that example, because a lot of the test is not written by me. Part of it is, but a lot of it's written by the author. And a large part of it has to do with naming compounds. When you look at it, and it gives you a choice, A, B, C, D, and say, what is the name of it? And he goes through that a lot. So this author does that a lot. Okay, so let's get to polymers here. And we found out that, and if everybody has this out, if you have that, that's the packet I gave you. You found out before that two kinds of polymers. One's the addition polymer, but that requires that you have double bonds. So if you have monomers that have double bonds, then a catalyst can actually break that double bond and form a bunch of single bonds, and it can actually hook things many long chain. A chain could be very, very long of carbon, and that would be an addition. And the good news about that is you don't lose any atoms, okay? Now the second kind, which happens a lot in nature, is this one called a condensation polymerization. And why use the word condensation? Because most people think of condensation as water, don't they? And most of the time, especially in the biological world, they'll take an H. So an enzyme will take an H off of one monomer and an OH off another, and it will dehydrate it. So you could call it dehydration. Some biology books will say dehydration synthesis. Chemistry books tend to say condensation polymerization. Same thing. Anyway, so you'll see this a lot in nature, and there is water. I think you did the condensation. Parker, did you do the condensation? It wasn't just water. It's other small molecules. Do you remember another small molecule that might have been removed?
Carbon?
No,
it wasn't carbon. It was.
I think it was like HCl, wasn't it?
I wrote down as a note. It was.
Was it HCl?
I think you're right.
So that means that some enzyme took an H off of one monomer and a Cl off another and formed a little molecule called HCl. All right. So what I want to show you is do you remember, let's see if you remember this. In biology class, if you remember from 9th grade, what do you remember as far as, here, let's try this. What were the 4 elements? What were the 4 elements found in the most abundance in living things? Does anybody remember those?
Carbon, hydrogen, oxygen, nitrogen.
Okay, and that's CHON, isn't it?
Oh, hydrochloric
Okay.
acid.
Yeah, HCl.
Yeah, HCl.
That's right. Okay, now, what are the 4 types of organic we know there's a lot of water. That's inorganic. There's a lot of water in living things, but the inorganic, that's inorganic. What were the 4 types of organic compounds found in most living things?
The 4 what?
Not the 4 elements, the 4, what 4 types?
Water's in there.
That's inorganic, though.
Oh, interesting.
And,
Is it sugar?
Carbohydrate.
okay, so, all right, proteins. It's not organic because no carbon, you know, so it's inorganic. Okay, now, so proteins, and we know proteins are definitely a polymer. And what is the monomer? Amino acids. We're going to talk about that a little bit today. And what is another one? That's right. Now, lipids, oh, by the way, I forgot to tell you this. Proteins are made of carbon, hydrogen, and oxygen, and nitrogen, mostly, okay? Lipids are fats and oils, and they contain carbon, hydrogen, oxygen, but they have a whole lot of hydrogen and only a little bit of oxygen, very little. And they're not really polymers. They're not really polymers. Okay, now what's the other one? And you're in biology, so you probably remember these. Is anybody who remember? right, you know one? What was another one?
No, come on, you should know.
Well,
I'm just kidding, I don't know.
okay, what's another one? Okay,
Oh, we did a little Mission stop too.
and that is,
Slipped out my hand.
those are polymers, but what's the monomer of a nucleic acid called?
Nucleus.
What?
Nucleotide.
Just saying what she said.
And we're going to talk about that too. What does that look like? And there's one more that is also a can or cannot be a polymer. And you want to go 4 for 4? Okay.
Are we eating?
Yeah, dude, why aren't you saying stuff?
Now, carbohydrates are your sugars and starches, but they are polymers. Some of them, most of them are polymers, and the monomers are sugars.
DNA
I
is
can't wear that anymore.
nucleic acid, right?
Okay, so one more time. Protein's a polymer, and the monomers are amino acids. So a protein is a long chain of amino acids that put together as a polymer. These are not really polymers. They do contain carbon, hydrogen, oxygen, but they have a whole lot more of hydrogen than oxygen. Nucleic acids contain C, O, N, and P. They actually have phosphorus in them. And if you remember DNA, they have a sugar phosphate. And so that's that one. And then carbohydrates have these three, but almost always in a 2-to-1 ratio of hydrogen to oxygen. So your famous glucose, C6H12O6, see how it's CHO in a 2-to-1 ratio here? Here's table sugar, C12H24O11, CHO, and here's your, almost a 2-to-1 ratio there. Okay,
Why is table sugar different from glucose?
okay, we're going to talk about that, but when we get to this one, you're going to find out that this is called a simple sugar, called a monosaccharide, and this has to be a double sugar, a disaccharide.
For a couple of announcements. One is Dialectic Girls, if you're on the soccer team, please wear your practice shirts today. Dialectic soccer players, wear your practice shirts. The other is club leaders, please make sure that you have something planned for your club on Monday. Thanks.
Wait, do we need to find something?
And then also, so this is like a double sugar, and then you're going to find out that you can do this. You can put a lot of sugars in a long chain, sugar, and this is where you call it a polysaccharide.
And that's not a.
Anyway, that is a polymer, okay? So of these, the ones that are polymers are these. All right, you ready? So let's go back over here, and we're going to find out that, let's start with proteins. Proteins are long chains of amino acids, all right? Those of you who go to school, if you're in science, chances are, if you're in any kind of medical field or biology field, you will be taking biology and chemistry probably at the same time, if I had to guess. And you'll actually find a lot of chemistry in biology. The first 3 chapters are probably going to be mainly biochemistry, like this. And so it actually helps to know chemistry pretty well. This right here is a long chain of amino acids called a protein. So what I'm going to show you, I did this try yesterday, they have different levels of protein structure. And one of them is called the primary sequence. And remember those little pop beads I had over there? And the pop bead says, what sequence are they in? And so we'll start, let's come over here. Here we go. Here's page 1025. It says, in this section, we'll consider a class of natural polymers, the proteins, which make up about 15% of our bodies and have molecular weights, molar masses that range from 6,000 to 100,000 grams per mole. That means some of these are tremendous polymers. They're macromolecules. Get that? Okay. Here's what I was talking about, that word. You said you hadn't heard that before, but Some of them are structural proteins, and they make up structures. They make up the cell membrane. They might make up the membrane of the nucleus. They might make up part of the ribosome. DNA has, chromosomes have protein in them. They might be structural. Another kind of protein it could be is called like an enzyme, and they act like catalyst, and they're the catalyst of the world. So there's two kinds of proteins mainly. One's for structures, one's for enzyme, for being catalyst. All right, and it says here the building blocks are amino acids. Okay, here's some good and bad news for you. Here we go. If you take a look over here, this page, this is what you're going to be doing for Tuesday. Now, of these, imagine the protein that makes a fly wing go like that, or the protein that makes up the hoof of a cow, or the protein that makes up your hair or your skin. They're all different. They're all different, okay? Because so there's millions of proteins. But imagine you were the first person to discover, well, I analyzed this, I analyzed this. I've been analyzing 200, 300 different things. And you know what? I've only found there's only about 20 building blocks. These millions of proteins, they're all made of about 20 different building blocks. And those things are called amino acids. That's amazing to me. That the protein in an ant's leg and my protein that makes my arm go like that, I have the same amino acid, but not the same number of each. Okay, so what I'd like you to do is, I'm going to show you this is a little easier than you think it is. I want you to pick 5 of these and 5 of these. Now I can tell you right now that I'll take this one as extra credit, and I'll pick this one as extra credit. So I think those are the hardest ones, but you can pick any 5 you want, and on Tuesday, I'm going to ask you to draw the structure of 5 polar amino acids and 5 non-polar. So let me show you how to make it a little easier for you, okay? If you look over here, every amino acid has something in common. They have a central carbon, so we start there. They all have a hydrogen right here, all the same. They all have an amino end, and they all have an acid end. Amino acid. Everybody get that? And the only thing that makes them different, of the 20 different amino acids, the only thing that makes them different is whatever's sticking right there. And they call that an R group. Now, let's take a look at this guy here. And I would definitely memorize this one. I used to be in high school. Okay. And what's the R group for glycine? It's just a hydrogen.
It's just
Okay. Now, if you look up here, these are called the non-polar group. You'll see up here, there's nothing here that would make it polar. So it's going to be non-polar. All right, what's the next one? I'd probably do this one. What's the R group here? It's a methyl group, isn't it?
Yeah.
And that's alanine. Okay, I'd probably do that one. This was a little more complicated. I probably wouldn't, if I had to memorize 5, I probably wouldn't pick that one. If you like it, you can pick it. All right, let's go down to, let's go down here. So here, isoleucine, what is that right there?
Oh,
So we have, see that? So here it has a little branch, here. And again, nothing there would make it polar. So it's called a non-polar amino acid. And here's one, methionine. Now, I didn't mention this, but look, that amino acid actually has sulfur in it. Not very many amino acids have sulfur in them. And this one, that one's a pretty good one to learn, leucine, and it goes like that. And again, though, look at the ends here. There's nothing really here to make hydrogen bonding. Okay, and here's the last one, valine, right there. And again, if you felt like doing an extra credit, I'll put that on there. And if you miss any others, you can get some extra credit points for that. Now let's take a look at the other amino acids down here. And these, I want you to look and say, what is it that makes it polar? So let's take a look at the simplest one here. This is CH2OH. What makes that polar?
It's got an OH in the end.
Right, because when they form a bond, it's not an equal bond, so you get that plus and minus nature, right? So no wonder this is a polar amino acid. Okay, that's called serine. Let's do another one. Look at this one. This one has a benzene ring in it. All right, but look at the end of it. That's going to make it polar. Okay, let's try this one. Look at this one here, glutamine. Now,
It's a nice guy.
remember that when oxygen and hydrogen form a bond, it's always polar because it's uneven electronegativities. Nitrogen always wins. It's just like, it's kind of like oxygen. It can take the electrons and pull it in there. So nitrogen, hydrogen, oxygen, hydrogen are going to be polar. So you can tell that's going to be polar there. Here's one. That's not too bad one to memorize. Lysine. Okay, there we go. That's polar. Arginine. There's one. Cysteine has sulfur in it, like that. And then here's one. Here's an OH group, aspartic acid. Now, I'm going to find one. Let me show you one here. Here it is. If you've been in biology class, if you've been in secondary biology, it's possible you haven't gotten to genetics yet. Have you done any genetics?
Yeah.
We've done some genetics.
Okay. So one thing that's interesting is that of all the amino acids, and all your proteins have amino acids, but some humans have trouble with this amino acid here. And it's called phenylalanine. And it's a genetic thing. And it's so, here's what happens. If they spot it early or they know ahead of time, all they have to do is don't give the baby any proteins that have phenylalanine in it for the first, 8 or 9 years. And then everything will be normal. But if you don't know anything about it, and you just feed them normal food, and as they get 2-year-old, 3-year-old, then you can actually hurt their brain and they can become retarded. Even though they shouldn't have been. They shouldn't have been. You can actually hurt their brain to the point where they have mental deficiencies.
By doing what?
By letting them eat foods, proteins that have phenylalanine in them. Now, you don't know which ones, so if you have a child like this, you're going to have to do a lot of research, and they'll say, this is fine, this is not fine. Sometimes you go to the baby section of a store, it'll say, contains no phenylalanine, and that's for these people. Now, I'm going to tell you why every single baby is tested for that, because it's that one genetic thing. If you just tell the parents, don't let them eat phenylalanine, everything's going to be all right. So they test them all, even though it's rare. And it's called PKU, phenylketonuria. Did you guys talk about PKU? Okay, so PKU, phenylketonuria, is the case where if you give them a lot of phenylalanine in the protein, and you just think it's normal food, then you can cause their brain to be messed up. And so we actually test every single baby for PKU because it's such a simple way to fix it. And after they get to a certain age, then they're actually okay as far as eating foods.
What are some foods that are?
I'd have to look that up. I haven't taught biology I used to teach biology for a long time.
Where'd you teach that?
And, I started in Indiana, I taught there. And then I came here and started teaching at Grimsley High School. And then I actually was here. I taught biology and advanced biology and honors biology here.
You were early?
Yeah.
I didn't know that.
You can go walk in the building, you'll see my name on the plan.
Remember, he's told us.
Only because I stayed there more than 20 years. But so, not that I was great. Well, I was all right. That was all right. But anyway, yeah.
That was
That was okay. Now I'm embarrassed. Okay, yes. All right. Well, anyway, so I want you to go home and say, here's 5 I like. I like those 5. And what I'll do is I'll actually draw this part for you. See that? I'll draw this part for you. You just have to know when you put an H there, you say glycine. When you put a CH3 here, you say alanine. There's 2 right away. All right? And then you have to pick 5 of these. So I'll ask for 5 of these. It's not till Tuesday. 5 of those, 5 of those. Okay? Now let's take a look. This is what I remember. I had never heard this at all in high school, and I went to college, and boy, my biology teacher, this was big. This was big with them. And that's over 45 years ago. And take a look at this here. Do you ever see this amino acid here? How do I know it's amino acid? Here's my central carbon. Here's a hydrogen. Here's the amino group. And here's the acid group. Remember, the acid group is COO, isn't it? C-O-O-H. All right, and here's what happens. In nature, when the cell wants to make a protein, there's this group of enzymes. And some enzymes know how to take an OH off of the acid group and an H off the amino group. Everybody see? Now, look at this. Wait, stop. Look, right here. Okay, here. Okay, so that is going to form water, and that's called, in biology, they call it dehydration synthesis or condensation polymerization, I think called, right? Ready, so you take a water off, but now, so what? So what if you took a water? Why would these two things want to form a bond? Well, see this guy right here, this carbon? He wanted to form 4 bonds, didn't it? 1, 2, 3, and you just remove this. So that carbon wants to form another bond, doesn't it? And this nitrogen wants to form 3 bonds, 1, 2, 3, but you took the hydrogen off, so the nitrogen wants to form another bond. And so they call that, when the carbon comes over and forms a bond with the nitrogen, where the amino group of this is making a bond with the acid group here, they call that a peptide bond. And then, therefore, if you do that over and over and over and over again, they would call that a polypeptide chain. Get that? Now, it's almost a protein. It's almost a protein, but it's not. It hasn't been folded up into its final shape. So right now, it doesn't work. But with the primary sequence of amino acids, that is the primary structure of a protein. Now, I want to show you. Okay, here it is. Fancy word. Peptide linkage means a peptide bond. Okay, a fancy word there. And a long chain of that would be a polypeptide, another fancy word like that. Okay, so how long can it be? Well, some books will say the shortest protein they know of might be around 80 amino acids long, but the other ones might be 600 amino acids long. So the protein sequence, you can have small, like 80, to make a protein, or even 600 to make a protein. Now that always amazes me that God made a structure, an assembly line, that says DNA tells the RNA, bring this amino acid, now this one, now this one, now this one, now this one. And you have to do that 600 times and not make a mistake. 600 different amino acids and put them in the right sequence, and then if it folds up right, it'll work. And that's amazing to me. Now, does it happen perfectly every time? No. And so sometimes you have the DNA may be okay, but somehow the way the message is read, they might make a mistake. And sometimes one amino acid, just one, out of order, can cause that person to, they at least lived, but they're going to have some kind of disorder. You might have hemophilia. They might have sickle cell anemia. They might have, they can't digest milk, okay? It could be, there's a lot of them that are simply a one amino acid mistake. So I want to show you, I had, here's on this page here. Here we go, let's see. Okay, here we go. How many different proteins would be possible then? It says here, write the sequences of all the possible tripeptides composed of amino acids, tyrosine, histidine, and cysteine. Well, let's just call them A, B, and C, okay? How many ways could you rearrange these 3 amino acids? How many ways could you rearrange them? I could go A, B, C. How else? I'll change the second one. A, C, B.
ACP.
Now, that's all I can do with having A as the first letter. Now, what about the next one? What if I want to have B as my first letter?
Good afternoon, Cowell Academy. My name is Isabella Marengu, and I am in Miss Cheney's 3rd grade class. I will be doing the prayer today. Bow your head and let us pray. Dear Lord, thank you for this day. I thank you that we had a good week, and if there's anything that is worrying any of your children, I pray that you will help them put their trust in you. Dear Lord, I pray for peace all over the world, especially in Ukraine. I know that you keep us safe every single day and that your love never dies. Bless all the students, teachers, and staff members of our school. It is in Jesus' name I pray. Amen. Now, one of my classmates, Carson Hicks, will read Philippians 2:14-15.
Do everything without complaining or arguing, so that you may become blameless and pure children of God, without fault in crooked and depraved generation in which you shine like stars in the universe. Have a great weekend, Caldwell.
That's so nice. That's so nice.
I knew he was gonna say. And he was like, have peace through all the world. I was like, he's gonna say something about Ukraine. So
All right, let me go ahead back here. So I've already exhausted all the ones that start with A. So I'm going to start with B. Well, BAC and BCA. Then I start another one, start with C, and I have, there's your chances. Now, so even though you could do it this way and figure out 6, there's another way of doing it, and it's called factorial. Have you heard of factorial?
Yes.
So 3 factorial is 3 times 2 times 1. And that tells you that there are 6 ways that you could arrange 3 things without repeating themselves. Now imagine this. We know there are 20 different amino acids, right? How many ways can you arrange 20 amino acids in?
10 to the 18th power.
Right. So that'd be 20. It'd be 20 factorial, right? That'd be 20 factorial, right? And you know what? Wait a minute. But that's only if it's 20 amino acids long. And we already know there are proteins that are 600 long. So imagine this number. If that's how we arrange to 20 by themselves, then the number is staggering. The number of sequences you can have are staggering. So I'm sure there are proteins in some animal or some plant somewhere in the world that we've never ever analyzed and never knew the sequence of those amino acids. And who knows, maybe that's a special sequence that could be the cure for something. That's why some people do that. They actually will look places that no one else has looked before and try to analyze. That's why they think if you're destroying a lot of the rainforest, for example, a lot of these, it has one of the highest diversities of any place on the planet. And so if you're destroying this all the time just so you can make money, there could be so many animals or plants that will never exist again. And God could have put those there so we could discover that one chemical that might be the cure or something. It might be there. Okay, so I'm going to show you one more thing here. First of all, the primary structure we talked about is just a sequence of amino acids, which can be lots of sequences. Now, the second structure is called the secondary structure. Once you get this long chain, remember the R groups, some are polar and some non-polar? They actually start attracting each other, and they start bending themselves into, and the two main shapes they can get into is a helix or a pleated sheet. And so here's what happens. In some proteins, they can actually bend themselves like this, and the amino acid chain, look at me, can be like a spiral, a spiral. Now, the other thing is that they can actually form sheets, and then they bend like that. It's like you take a piece of paper like an accordion, and that's called a beta pleated sheet. But I had 2 pictures here I want to show you. Page 974. Look up here, okay? Okay, so 974, I want to show you a protein, and this is called a heme group, hemoglobin. Have you ever heard of hemoglobin? Okay, take a look at hemoglobin. Now, hemoglobin is this structure here, and one thing I've always thought was fascinating was that there are lots of hemoglobin molecules in your red blood cells. And hemoglobin is that one chemical that knows how to grab onto oxygen and deliver it to the rest of your body. Because when I breathe, I have cells here in my toes. They're not exposed to my lungs, but the lungs make sure the oxygen get into the hemoglobin, and then the red blood cells carry that to every cell in my body. And even if it can't touch a cell, if you remember the word diffusion, remember that? It can give oxygen to this cell, and then the content of this cell has a higher oxygen, and it'll pass to the next cell, and every cell in my body can get oxygen that way. Now, look at this right here. That's a weird structure. It is organic, but what is this? These are nitrogens, and what's in the middle? And no, what's in the very middle?
Not hemoglobin.
See, that's so strange. You have this organic molecule, but what's in the middle? Wait, Mason, okay? What's in the very middle?
Is that iron?
It's iron.
Wait, you tell me that?
And so everybody who knows about, hey, you need iron, why do you need iron? And because every one of your hemoglobin molecules has an iron atom in the center of it, doesn't it? And if you don't have enough iron, then you can't make enough hemoglobin, and you'll be anemic. You might not be able to make enough red blood cells, and you'll have anemia. All right, now look down here at the bottom. I'm not trying to tell you that these are identical. They're not. They're not identical, but they are similar,
What does that look like?
except for this. See that tail? They don't have a tail up here. But look at this. This is found in many, many plants. So this part here is, I'll get there in just a minute, is similar to that, right? But look what's in the middle of this one. What's in the middle of a chlorophyll molecule? And I thought that's so interesting that a very, very important chemical that has to do with mammals, this site has a metal atom in it, and then this one has a metal atom in it. Yeah, go ahead.
So say you have anemia, you're an anemic. What does that mean for your life? Like, what is that?
Okay, so,
Change.
all right, so if I were an athlete and I were anemic, how much oxygen can I deliver to all my muscles and things? I'd have trouble being a top-notch athlete for sure. I might run out of breath and get tired very soon. The people who have sickle cell anemia, very, very few of them are athletes. They want to be, but they can't. Their red blood cells, because of its genetics, instead of being a nice little disc, they actually are like sickle shape, like a curve, and they get clogged up in the arteries. And so if they're not careful, they have lots of pain. They can't deliver enough oxygen, so they have to be careful about how they exert themselves. So another thing is, for a lot of people, they tend to look more pale because you don't have as much red blood cells. But that's minor compared to, hey, I can't get my breath, I can't get enough oxygen, I can't walk more than half a mile or something like that.
I'm gonna make a theme announcement
Yeah,
for the bench.
okay. All right, so I want to show that to you, and there's another one to show you.
Ask my mom.
Let's take a look right here.
No, you ask her.
Now here is one. I wanted to show you that the primary structure of proteins are the sequence of amino acids. Let me finish, okay? The primary structure, I already talked about it over and over again. It's a sequence of amino acids, which can be huge. And then the second one, they either form themselves either into a spiral or a pleated sheet, and then they have a tertiary structure. The third level of protein structure is when, even though you might have spirals and pleated sheets, it will then fold itself into its final shape, and it might look like this. So this might be an actual protein in its final shape, like that. Now, I know I've had a lot of biology. I've done a lot, so I know what this means, that's important. And I know that if anything in my environment causes that to be out of shape, it's not going to work right. So no proteins are very sensitive to changes in pH and things like that. Remember we did buffers?
Yeah.
Every time I did some research.
What's up, everybody? God, it is good today because he gave us a home baseball game. So for the theme for that game is going to be Blackout. Hope to see y'all there at 4:30.
Now, if you don't tell the people until now, did they already know to wear blackout today?
I think he just came up with it while he was sitting there. He was
But the good news, we get out at 2 today, so therefore they can go home. They can go home and wear blackout. Okay. All right, one more. Let me show you real quickly here. Notice that this is found here, and there's another. I don't want to get into this, but the quaternary structure means sometimes you'll have one of these and something else, and then they'll form another complex thing. And so biology, I don't know where you stand with evolution or things like that. I don't know where you stand, and that's okay. You have to work that out by yourself anyway. But this is so complex, and yet it took very little to make this not work. And yet every one of my cells might have one of these, the red blood cells, for example. So just amazing. I'm going to show you, here's a picture I was talking about. One amino acid out of order, one, and it's passed on with genes, it's genetic, can cause you to have these kind of red blood cells. These are sickle cell, and these are irregular. These are regular red blood cells. And these go through arteries and capillaries, just tumble. They don't have any trouble going through your blood vessels, but these get clogged up and they have trouble. And so, have you ever met anybody who was sickle cell? Okay, you have? Okay. If I had a chance, was it a black person? Okay. It just happened to be in the black population. This would be talked about in almost every family in the black population. You probably never heard about your house. But the percent of black population that have sickle cell is way higher than it is in your population. for the white population, cystic fibrosis is higher in the white. You'll never hear about that. You probably never heard that either, have you? Oh, you have? Okay, so that one is much higher in the white European background there. There's another one, and I'm almost done here. Give me 30 seconds. There's another one. If you're a Jewish population, they would talk about Tay-Sachs disease, and that's another blood thing. So anyway, there are different things that are connected to DNA, which are connected to proteins, which are connected to cells, and it's a great picture. All right, here's what I'd like you to do. I won't see you on Monday because the gold schedule, I won't see you till Tuesday. So when you get back, I want you to say, I'm going to memorize 5 polar and 5 non-polar amino acids, and then I'll see you, okay?
So 10 total, 10.
Don't forget, I've been taking my book home. You might want to start reading. If you want to wait till Monday and read, you can do that.
Probably about 7.
I'm
Thank
hoping
you,
that
Steve.
some of the stuff we do in here, you've at least heard, is it connecting a little bit?
Yeah.
She's gonna tend to keep it all in the biological realm, maybe, but maybe not as much in the chemistry. Maybe.